Main Biotechnology Services is an antibody discovery company dedicated to bringing unique and compelling antibody targets to market. By leveraging 20 years of expertise in hybridomal development sciences, we have successfully added a number of new and unique antibody products to our catalog. In order to expand the current offerings of 5 monoclonal antibodies specific for human osteopontin, R&D scientists at MBS developed a panel of antibodies targeting a functionally important site, dsvvyg, of osteopontin. MBS is proud to announce the availability of this unique panel of antibodies specific for osteopontin, which is an increasingly important biomarker for many cancers and other disease states. The goal of the project was to develop antibodies which specifically targeted the dsvvyg site on osteopontin, often referred to in the literature as the cryptic site of the molecule. Besides being heralded as an integrin receptor when the molecule is cleaved, the cryptic site of OPN is also compelling because it is neither phosphorylated nor glycosylated, unlike the rest of the molecule. The lack of post-translational modifications on the cryptic site makes it conducive for antibody binding. To this end, MBS designed and produced a peptide immunogen incorporating the amino acids DSWYG, and this was conjugated to a KLH using cross-linker design to reduce steric hindrance. Similarly, a BSA conjugate was prepared for screening. The hybridoma project resulted in 28 clones specific for the peptide, scrubbed for any clones which were specific for linker molecules. The 28 clones were purified from supernatant via the MBS multi-pure proprietary technology and analyzed by an indirect ELISA against the BSA conjugated peptide, the E. coli produced recombinant osteopontin, both end-term fragment and full length, as well as native osteopontin purified from human breast milk. An SDS page of the purified osteopontin reveals that it appears to be primarily full-length OPN, although it is acknowledged that glycosylation of OPN can interfere with protein staining. An isoelectric focusing gel was run on all 28 clones. 9 buckets of isoform patterns emerged, and a representative clone was selected from each for further study. The data indicates that the clone's ability to recognize the cryptic site by indirect ELISA varies and is dependent on the source material of osteopontin. Not all clones were able to recognize by indirect ELISA recombinant and/or native OPN to the same degree. MBS currently offers 5 monoclonal antibodies, MAB193 through MAB197, to human osteopontin. 4 of them are specific to the N-term fragment and 1 clone is specific for the C-term fragment. A study was conducted to determine how well the peptide clones could pair with the currently available monoclonal antibodies to detect native and recombinant full-length OPN by ELISA. Using monoclonal antibodies, MAB193 through MAB197 as capture, and the peptide-specific clones as tracer, it is clear that there is a significant difference between the ability of each pair to recognize the recombinantly expressed osteopontin versus a native source of human osteopontin in a sandwich format. The E. coli expressed osteopontin does not account for naturally occurring post-translational modifications, nor can it be assumed that the recombinant molecule has maintained the native conformation. As a result, the ability of the antibody to bind recombinant OPN and not native indicates that the epitopes of these clones may incorporate areas encumbered by post-translational modifications and/or target areas that are folded differently. The takeaway message from the data speaks to the significance of using native OPN, or an OPN that has been expressed in a mammalian system as the antigen in the ELISA assays. Of note, MAB193 pairs very well as capture with many of the peptide clones to measure native OPN. In the reverse assay format, that is, using the peptide clones as capture with MAB193 through 197 as tracer, Again, we see the discrepancy between the use of a native OPN and a recombinantly expressed full-length OPN. The sandwich ELISA data reveals that MAB193 pairs well with clones 14C08, 15C02, and 16A11 in either ELISA sandwich configuration. These clones have been selected for stabilization, production, purification, and characterization. The resultant antibody product will be made available for sampling. In studying the effects of OPN on the behavior of cancer cells, often the researcher performs cellular adhesion assays to ascertain whether or not an antibody bound to OPN can inhibit the cancer cell's ability to adhere. Using a breast cancer cell line MDA-MB-231, 3 clones, 09B05, 16A09, and 16E12, have been shown to exact this property using recombinant N-terminal and full-length osteopontin. Future work includes repeating the assays using native osteopontin. All 3 of these clones are undergoing stabilization, production, purification, and characterization. The purified antibody will be made available for sampling. To meet the needs of osteopontin researchers worldwide, MBS embarked on a hybridoma development project targeting the known cryptic site of osteopontin. To this end, a peptide immunogen targeting the dsvvyg amino acid sequence of the peptide was designed and produced for the project. 28 resulting clones emerged, which were narrowed down to a panel of 6 clones of strong interest based on ELISA data as well as cell adhesion assay data. All 6 of these clones are specific for native OPN by ELISA, and 3 are able to affect breast cancer cell adhesion. This represents a new and unique cohort of osteopontin monoclonal antibodies which are available to the market. This study also revealed the importance of using native OPN or potentially a mammalian-expressed OPN in research to accommodate for the prevalent post-translational modifications occurring in the native form of OPN. Additional studies currently underway include a Western blot analysis using native purified OPN against all 6 of the selected clones, as well as cell adhesion assays using the native OPN. If the cryptic peptide is linear, the Western blot data will shed light on the integrity of the purified native OPN by immunostaining full-length and/or OPN fragments containing the DSWYG, indicated by molecular weight of the bands. Additionally, the ability of these clones to inhibit cell adhesion by binding to native OPN will be of significant value to cancer researchers. Results of the extended studies will be made available at a later date. Please contact sales to access samples of this unique panel of osteopontin monoclonal antibodies.